Phagocyte ingestion of complement-tagged pathogens is mediated by receptors for the bound complement proteins. The most important action of complement is to facilitate the uptake and destruction of pathogens by phagocytic cells. This occurs by the specific recognition of bound complement components by complement receptors, CRs, on phagocytes. These complement receptors bind pathogens opsonized with complement components. Opsonization of pathogens is a major function of C3b and its proteolytic derivatives. C4b also acts as an opsonin, but has a relatively minor role, largely because so much more C3b than C4b is generated. The best characterized is the C3b receptor CR1, which is expressed on both macrophages and polymorphonuclear leukocytes. Binding of C3b to CR1 cannot by itself stimulate phagocytosis, but it can lead to phagocytosis in the presence of other immune mediators that activate macrophages. For example, the small complement fragment C5a can activate macrophages to ingest bacteria bound to their CR1 receptors. C5a binds to another receptor expressed by macrophages, the C5a receptor, which has 7 membrane-spanning domains. Receptors of this type couple with intracellular guanine nucleotide binding proteins called G proteins, and the C5a receptor signals in this way. Proteins associated with the extracellular matrix, such as fibronectin, can also contribute to phagocyte activation. These are encountered when phagocytes are recruited to connective tissue and activated there. C3a, which has inflammatory activities similar to those of C5a, although it is a less potent chemoattractant, binds to its own specific receptor, the C3a receptor, which is homologous in structure to the C5a receptor. 3 other complement receptors, CR2, CR3, and CR4, bind to inactivated forms of C3b that remain attached to the pathogen surface. Like several other key components of the complement, C3b is subject to regulatory mechanisms and can be cleaved into derivatives that cannot form an active convertase. One of the inactive derivatives of C3b, known as IC3b, acts as an opsonin in its own right when bound by the complement receptors CR2 or CR3. Unlike the binding of IC3b to CR1, the binding of IC3b to CR3 is sufficient on its own to stimulate phagocytosis. A second breakdown product of C3b, called C3DG, binds only to CR2. CR2 is found on B cells as part of a co-receptor complex that can augment the signal received through the antigen-specific immunoglobulin receptor. Thus, a B cell whose antigen receptor is specific for a given pathogen will receive a strongly augmented signal on binding this pathogen if it is also coated with C3DG. The activation of complement can therefore contribute to producing a strong antibody response. This example of how an innate humoral immune response can contribute to activating adaptive humoral immunity parallels the contribution made by the innate cellular response of macrophages and dendritic cells to the initiation of a T cell response. The central role of opsonization by C3b and its inactive fragments in the destruction of extracellular pathogens can be seen in the effects of various complement deficiency diseases. Whereas individuals deficient in any of the late components of complement are relatively unaffected, individuals deficient in C3 or in molecules that catalyze C3b deposition show increased susceptibility to infection by a wide range of extracellular bacteria. And that's phagocyte ingestion of complement-tagged pathogens is mediated by receptors for the bound complement proteins. Have a great day.